talking about meiosis and sexual reproduction. So, when an organism is going to reproduce, it does so by the creation of gametes. And those are sperm cells and egg cells. And sperm cells and egg cells each have only half of the genetic material a normal cell would have. So in humans, a normal cell in the body has 46 chromosomes, or 23 pair. Well, a sperm cell and an egg cell only has 23 chromosomes. Alright, 23, half the number. And that is because to make a new life, when the sperm and the egg come together, the 23 from the father and the 23 from the mother come together to make the new zygote, the new individual, the offspring. So meiosis is the process by which we make gametes. So meiosis, we make gametes. We make cells that only have half the total number of chromosomes. Alright? So when we're talking about meiosis today, that is what we're going to be talking about, the process by which we make a cell that only has half of the total normal number of chromosomes. Right. So, just a little bit of a review. Humans have 46 chromosomes. They have 2 copies of each chromosome in the cells that make up their body. So, for example, here's like a human karyotype. And a karyotype just means a picture of all 46 chromosomes. They're separated into 23 pairs. 22 numbered pairs, and then an X and a Y chromosome, which is the 23rd pair. And you can see that for each number, a human has 2 copies of that chromosome. Now, they're not genetically identical, but they contain the same genes, you know? So, if we have 2 of each of these 23 pairs, that's 46 in general. Organisms that are made up of cells with 2 copies of each chromosome, like humans, see we have 2 copies of each chromosome, are called diploid. Or cells that have 2 copies of each chromosome are called diploid. Di meaning 2, ploid meaning chromosome. So most human cells are diploid. They have 2 copies of each chromosome. The 2 copies of the same chromosome are called homologous chromosomes. So, for example, these 2 are homologous chromosomes. These 2 would be homologous chromosomes, these 2. So, the 2 copies of the same chromosome are called homologous chromosomes. When diploid organisms reproduce, each parent, their mother and their father, each contributes half of their genetic material to their offspring via their sperm and egg. Therefore, the sperm and egg, the gametes, remember, are haploid. They only contain 1 of every chromosome. So, if these are normally in most of our cells that are diploid, sperm and egg cells would have only 1 copy of each chromosome. They would be haploid. They would only have 23 chromosomes. So I'm going to color out 1 of each chromosome, and this, what we're going to be left with, is what a gamete would have. So they would lose 1 of these copies of each chromosome. So this might be a sperm cell that has 1 copy of each chromosome. And then when it combined with an egg cell from the mother, the egg cell would have the other copy of each chromosome. So they'd come together, and the offspring that would be created by fertilizing the sperm with the egg with the sperm would have 2 of each chromosome. So meiosis is the process in which we can get a haploid gamete from a diploid cell. So we take a diploid cell and we basically make a gamete that has half the total number of chromosomes. So 2 haploid gametes come together to make a new diploid organism. So for example, if this is my sperm cell, and it has 1 copy of chromosome 1, copy of chromosome 2, copy of chromosome 3, so this is my sperm cell, and it's going to go and fertilize my egg cell that has 1 copy of chromosome 1, copy of chromosome 2, copy of chromosome 3. We're going to get out a zygote that has 2 copies of chromosome 1, 2 copies of chromosome 3, 2 copies of chromosome 3. We're going to combine 2 haploid cells, and we're going to get a diploid organism. Perfect. This is what happens in human. Here's another picture of it. This is how we take these 2 haploids and we make a diploid or a new life form, a zygote. So now we're going to look at what specifically is going on in meiosis. Well, a lot of it is similar to mitosis, except there's 2 major differences. Meiosis only happens in the testes and the ovaries, so where the human gametes are made, or other animals where those gametes are made. So they're only happening in these specific places. Meiosis doesn't happen in any other place in your body except where gametes are made. Unlike mitosis, 2 rounds of cell division compared to just 1. So in meiosis, the parent cell divides once, twice, and creates gametes. But DNA is only replicated once at the very beginning. So it goes through something like mitosis once, where it duplicates DNA, splits into 2 daughter cells, but then those 2 daughter cells split in half again without duplicating their DNA. And that's how we get gametes. So each phase occurs twice. Prophase, metaphase, anaphase, telophase. They all occur twice. And we use little Roman numerals 1 or 2 to represent which prophase we're in. Prophase 1 would be the first prophase, prophase 2 would be the second. What we get out is 4 daughter cells, unlike mitosis where you get 2 daughter cells, with half the original number of chromosomes. So what we get out are haploid daughter cells from a diploid mother cell. And here's kind of an overview. Alright, so we have, maybe this organism only has 1 chromosome. 1 chromosome, 2 copies of each in the diploid cell. And it duplicates its genetic information. So now we have 2 copies of that homologous chromosome right here. Then in the first division, the homologous chromosomes, the copies of the same chromosome, separate. So after the first division, each cell only has 1 copy of each chromosome. And in the second division, those sister chromatids of the same chromosome separate, and we get 4 haploid gametes. Now, stages of meiosis. Right, now we're going to look at the first division. So prophase, metaphase, anaphase, telophase 1. First, the diploid cell copies each chromosome, just like in mitosis. So we go through S phase. Before mitosis starts, we copy our genetic information. We form those X-shaped chromosomes that are condensed. But during metaphase, homologous chromosomes line up together along the metaphase plate. This is different from mitosis. So in metaphase 1, the homologous chromosomes, let's say this is chromosome 1A, 1B, 2A in blue, 2B in red, 3A in blue, 3B in red. They line up along the metaphase plate like this. So homologous chromosomes line up across from each other. In mitosis, this would be 1A, and 1B lines up right next to it. So in mitosis, they line up right next to each other. In meiosis, they line up across from each other. Big difference, because then when the spindle fibers pull homologous chromosomes away from each other, the resulting cells only have one copy of each chromosome. So these resulting cells are already haploid. They have half the total number of chromosomes, just they have two copies of the same homologous chromosome. Alright. So how is division 1 different from mitosis? Well, like I said, in mitosis, the individual chromosomes line up. Mitosis, big difference. Metaphase, individual chromosomes line up in the middle. Like this, each individual chromosome. So here's like 1A, 1B, 2A, 2B, 3A, 3B. So one sister chromatid of each of the 46 chromosomes is going to the other side. So each new cell after mitosis will have one chromosome of each of the 46. In meiosis, during metaphase, homologous chromosome pairs line up in the middle. So just like in this picture, now they're not all N10, but all the 1 chromosomes, 1A on one side, 1B on the other side. Maybe 2A's on this side, 2B's on this side, 3B's on this side, 3A's on this side. So the pairs, all like numbers, line up so that when they divide, each new resulting cell will have 2 copies of the same A or B. So it will result in only having haploid number 23 of the 26, but we'll have 2 copies of each of those 23. Also in meiosis 1, when their chromosomes are lining up along the middle here, since they're paired up in prophase 1, something called crossing over happens. So during prophase of meiosis 1, when copies of homologous chromosomes are lining up together before metaphase, pieces from one of the homologous chromosomes can swap with a piece from the other homologous chromosome. So maybe this is my 1A chromosome, 1B. Right here, they kind of attach to each other and form something called a chiasma. And pieces of the 1B chromosome can switch with the 1A chromosome. This is called crossing over. It increases genetic variation because now I'm taking part of a different chromosome and sticking it onto another, creating genetic variation that wasn't possible before. When homologous chromosomes pair up, this is called synapsis. This is when the actual switching over occurs. So this chiasma forms during synapsis, and crossing over happens. This happens only in meiosis 1 during prophase. Something different from mitosis. Then meiosis 2 occurs. In meiosis 2, the 2 daughter cells created from meiosis 1 divide again without replicating DNA. This separation is exactly the same as mitosis. Sister chromatids separate, all of the chromosomes line up along the metaphase plate, and they get pulled apart, so each resulting daughter cell will have one sister chromatid of each 23 chromosomes. Alright? So, prophase, metaphase, anaphase, telophase 2, pretty much the same as mitosis. Essentially the same as mitosis. So, overall, let's look at what's similar and different. Here's some good diagrams going through mitosis and meiosis. Remember, first, during prophase 1, synapses and crossing over chunks of homologous chromosomes or switching in meiosis never happens in mitosis. 2. Metaphase 1. As you can see, homologous pairs line up. One pair on one side, one on the other, and they can be in any order. 1A can be here, 1A could be here. It doesn't matter. In mitosis, each chromosome is on the plate. Here, they pair up. When they divide, homologous pairs will separate. So we have a haploid cell already. And then we go through a second round of division. So we end up with 4 haploid daughter cells with 23 chromosomes. We end up, after mitosis, with diploid with 46. So, pay very much, and I would study this diagram in your book. What's the same in DNA replication? In both, it happens in interphase before meiosis 1. Difference: number of divisions, mitosis 1 division, meiosis 2 divisions. Synapsis and crossing over does not occur in mitosis. It does occur in meiosis. 2 diploid daughter cells result from mitosis. 4 haploid daughter cells result from meiosis. Mitosis happens in all cells to grow, repair, replace. Meiosis only happens in gametes. Alright. Lastly, 3 ways meiosis increases genetic variation. This is important to remember as we go into genetics. 1. Independent assortment. So each of the 23 homologous chromosome pairs can align one of 2 ways during metaphase of meiosis 1. So, this red chromosome could be on this side or this side, and it's totally random. At the same time, this red chromosome can be on this side or this side, totally random. Doesn't depend on what happened before. All the 1A, 2A, 3A don't line up on one side, all the 1B, 2B, 3B don't line up on the other side. It's completely random. That's why it's called independent assortment. And this results in 23 to the second total different options of how they could line up. A lot of different variation. Crossing over can occur, which swaps genetic information between chromosomes. It doesn't happen in the same predictable way every time. And third, random fertilization. So an egg cell can be fertilized by any sperm cell with any combination of genetic material that resulted from independent assortment and crossing over. So in total, 70 trillion different combinations are possible for each combination of any sperm and any zygote from 2 known individuals. Once you add into random mating, this becomes close to infinite different combinations. So if you think about all these things, you are truly unique. There are over 70 trillion different possibilities of how you could have come out as far as genetic information. Alright, that's it for today. Please let me know if you have any questions.